Hello everyone, my name is Alexandra Crespin, and I am going to present how viruses can mutate and how the novel SARS coronavirus 2 could mutate in the future. So firstly, what is a virus? A virus can be defined as a non-living infectious agent or particle that infects a host organism or cell. There is a limited number of species that a certain virus can infect, and this is called a host range. Viruses do not perform their own metabolic processes, and they cannot reproduce without first infecting a host. After a virus comes into contact with a host cell, the virus releases its viral genome, either of RNA or DNA, and infects the cell, allowing its dangerous genetic sequence to spread to other cells. The image depicted on the left shows the main parts of a virus. So how does virus, infection, and mutation take place? Many known diseases stem from a single viral particle. These diseases and illnesses include, but are not limited to, smallpox, the common cold, rabies, Ebola, and the novel coronavirus disease, known as COVID-19. Viruses are affected by evolution, just like plants, animals, and humans are. Subtle genetic changes brought about by evolution can result in virus mutations, which occur when an error occurs as the virus is transferring or spreading its genome. Mutation rates are higher for RNA viruses than DNA viruses. This means that viruses with an RNA genome are more likely to produce mutated virus copies. This occurs usually since RNA polymerase enzymes do not proofread the order of bases in the genome like DNA polymerase enzymes do. There are three main mechanisms of virus mutations. First, physical mutagens like X-rays or ultraviolet light rays can damage a virus's genome and affect the layout of nucleic acids. Second, natural behavior of the nucleic acid bases in the genome can result in genetic mistakes. Third, there are errors which can occur involving the possibly fallible enzymes which are supposed to build or correct the order of nitrogenous bases in the genome. Keep in mind that mutations to viruses pose a danger to affected species if they create a new antigenic strain of the disease, which can infect people or organisms that have already had the illness previously. What are the ways in which SARS coronavirus 2 can mutate? As an RNA virus, SARS-CoV-2 is more likely to develop mutations which can change the virus. However, there have only been slow changes so far. This virus has a proofreading enzyme which prevents some mutations and gives the virus a mutation rate slower than that of influenza or HIV, which are two other RNA viruses. One mutation, which has already been spread, is the D614G mutation. This mutation was on the gene encoding the spike protein, pictured to the right, on the 614th amino acid position, which is used to penetrate cells, and some scientists believe it created a more transmissible form of SARS-CoV-2 that was able to spread to different continents around the world. Mutations are easy to find, with the new access to the virus's genetic code from thousands of patients, but causes and effects of these mutations is difficult to research. Some possible mutations to the spike protein's receptor binding domain could make these proteins unrecognizable to antibodies, making the virus more difficult to fight. This is what mutations in SARS coronavirus 2 can mean for the future of the pandemic. Mutations are impossible to predict. There are a multitude of ways that this virus can mutate in the next few months or years, and scientists will be unaware of these mutations until they arise. But not all mutations are necessarily bad. Some genetic changes may weaken the virus and lead these viral cells to be more easily detected by a future vaccine. Regardless of which mutations can occur, scientists are monitoring SARS coronavirus 2 very closely and are preparing to tackle any mutations that pose a threat to the world's recovery from this pandemic.